afternoon, everyone, and welcome again to the BP Endure virtual seminar series, Champions for Diversity in the Neuroscience. And this is our last, but not least, seminar of this wonderful journey that we have had during this semester. And today we have the pleasure of having Dr. Diana Bautista, who is professor of the Department of Molecular and Cell Biology and the Helen Neuroscience Institute at University of California, Berkeley. And Dr. Bautista, I met her at Woods Hole because both at the time were co-directors of summer program. In particular, Dr. Bautista is currently the co-director of the neurobiology course at the MBL in Woods Hole, while I was at the spines course in as well that summers. And to tell you a little bit about her background, although she will talk a little bit on her in her talk, she received her bachelor's degree in biology and biochemistry at the University of Oregon and her PhD in neuroscience from Stanford with Rich Lewis, and was a postdoctoral fellow in physiology at University of California, San Francisco with David Julius. And then since 2008, she joined UC Berkeley. And regarding her research, Dr. Bautista studies the molecular and cellular mechanisms of itch, touch, and pain under normal and disease conditions. And her research has been funded by NIH since 2009, and her work has been recognized by numerous awards, including the 2014 Society for Neuroscience Young Investigator Award and the 2016 Howard Hughes Medical Institute Scholar Award, and more recently, the NIH Director Transformative Research Award. So thank you, Diana, for being with us, and we look forward to your talk.
All right, great. Thank you so much. Let's see if I could share my screen, if that will work. Let's see. Uh-oh. How's that?
Yeah, we see it.
Okay, thank you. So, all right, so what I thought I would do today is just tell you a little bit first about my path and how I got to be where I am today. And then I thought I would tell you a little bit about the graduate program at UC Berkeley. And then I would end with some research that my students have been working on. So, and of course, feel free to ask any questions. I'm happy to chat about any aspect of this. So, oops, let's see. There we go. So, oops, I grew up in the inner city of Chicago. I don't know if you can see my pointer over here on the northeast side of the city. I grew up in the same house with my mom, who was a single mom, and my sister, and my grandparents, and my great-grandparents, and my aunt. And we lived in one house all together. And I'm the first person in my family to graduate high school and to attend college. And I remember growing up, we were surrounded by all of these amazing schools that do really amazing research, University of Chicago and Loyola and Northwestern. And there's amazing science going on. But when I was in high school, I was really just focused on getting through high school and wasn't really sure what I was going to do after college. And I certainly never thought that I would end up being a scientist. And in fact, when I was growing up, I could only remember one instance where I even thought about science. And that was when I was about 8 years old. And I saw an ad for this chemistry set that showed that you could do exciting experiments, and it showed a little flame, then you could boil substances, and there were all these colored tubes. This is just a part of the ad. And the thing that really got me interested in this chemistry set when I must have been like 8 or 9 was it said in the ad that you could synthesize rotten egg gas to scare your little sister. And for me, that was really exciting because we shared a really small room that was probably just like a closet, and I wanted to get her out of my room. And so I asked Santa for this chemistry set, and on Christmas we open our presents, and I had this big box, and I thought for sure I was going to get the chemistry set, and instead Santa gave me this cut and curled Barbie beauty center. And really, my family thought it was really weird that I was doing science. We didn't know anybody who was scientists. All we knew about scientists was that they were, from TV, and they were crazy, and they were men, and they were trying to, point me in a direction that was more in their comfort zone, and trying to put me on track for, potential more realistic, what they viewed as a more realistic career. And everyone in my family, they're blue collar workers who work for some aspect of the city of Chicago. So after high school, I ended up going to art school, studying art for a year, and I was a really bad artist. And so I took some time off, and I really didn't think about science again until my gap years off of after high school, where I became involved with an environmental group that was studying dioxins in the Great Lakes, where I lived, and also that was fighting the siting of hazardous waste incinerators in low-income communities like the one that I grew up in, with promises of jobs and little discussion about the health consequences of these types of industries on the people in those communities. And this got me very interested in learning about science again, and thinking about epidemiological studies and toxins. And I started studying chemistry and reading different epidemiological reports. And eventually I went back to community college because I was more interested in art. In high school, I didn't take very many science or math classes. And so I went back to community college to see if I could actually do well and remember how to study and learn about chemistry in a formal way. And eventually I went back to the school, and I went to the University of Oregon in Eugene, where I was accepted and enrolled in their environmental science program that involves a combination of ecology and chemistry. But I was really lucky in that I found a lab to join very early on through the Federal Work Study Program. And instead of working in the cafeteria, I got a job in a neuroscience lab that was studying vision in fruit flies. And through having a wonderful mentor and working in an international diverse group of neuroscientists in Peter O'Day's lab, first making fly food and then eventually doing my own experiments, I became introduced for the first time to a career in research and academia, and I learned about graduate school and that you don't have to pay because I had a lot of loans. I had the federal Pell Grants, but I also had student loans to help pay, in addition to my work study, for my schooling, and I didn't want to take on additional loans. And also through programs like the HHMI undergraduate research program and the NIH MARC, I got to get paid during the summer to do research. And that really helped solidify my interest in research and also in neuroscience. So I applied to graduate school with a lot of help from these programs and my mentors. And I ended up going to Stanford for graduate school, and then eventually to UCSF for my postdoctoral training. And one thing that was really critical for my success is to have really inclusive mentoring, both as a graduate student and postdoc, and finding a mentor who can train me in not only how to be a scientist, but also how to communicate my science, how to develop professional skills to give a talk, to do networking, all of these things that I really didn't know how to do. And they also were very encouraging of me doing outreach work as a graduate student, a postdoc, because I felt like I had received so much mentoring due to outreach programs, and especially as a first generation URM, that I wanted to give back to my community and to reach out to other students, both at Stanford and UCSF. And they were really supportive in that. So in addition to learning as a graduate student how to study cellular communication and working on ion channels, I learned a lot about being a good mentor, and through example and also through outreach. And as a postdoc, I became very interested in ion channels and how ion channels allow for our sensory systems to become excited in response to different external stimuli. And so as an undergraduate, I studied how light gets converted into electrical signals in the Drosophila visual system. And as a postdoc, I looked at natural plant products in the brassica family. So isothiocyanates from mustard and wasabi, and different types of pungent vegetables, and how plants have evolved these chemical production systems as ways to target and repel mammals by producing these compounds that target our sensory systems and trigger pain and inflammation that prevents most mammals from eating them, although as humans, we often like the pungent plants like wasabi with our sushi. And it turns out that these compounds can activate sensory neurons that innervate the skin and the airways and the gut to trigger inflammation, and mimic endogenous compounds that are produced under really extreme conditions of disease or injury. And oops. And let's see, where's my slide? Oops, I seem to be missing a slide. And that got me interested in working on sensory systems in my own lab at UC Berkeley. And I'll just tell you a little bit about the department that I'm in, molecular and cell biology, and encourage you to either visit for the summer to participate in one of the many summer research programs that we offer to undergraduates, or to consider applying to graduate school. And so what is special about our department? Berkeley is a big school, but our department has a really nice culture of science and mentorship and education. We do, I think, really wonderful research, but also take training and mentoring very seriously, and also teaching. And so a big part of graduate school, in addition to research, is also becoming a teacher, becoming a mentor, and training undergraduates. It's really collaborative, interactive, and interdisciplinary. And we have many devoted scientists who span research from very fundamental basic science all the way to translational medically oriented research. And even though we're a basic science institute and we're not a medical school, one thing that's people find surprising right now is that we have more people who are currently studying mechanisms of COVID-19 than Stanford Medical School and UCSF Medical School combined. Let's see. We have over 90 labs in 5 different divisions covering all areas of molecular and cell biology, but without any boundaries. And so generally faculty teach and are associated, with biochemistry or cell and developmental or genetics, genomics, immunology, or neurobiology. But I'm very active because our research is very cross-disciplinary. And so my primary appointment's in cell and developmental biology, but we use genomics, we do neuroimmunology, and we also do a lot of molecular signaling and looking at ion channel structure function. So pretty interdisciplinary And these represent different divisions that students initially join, but then you're free to work in any of the 90 plus labs associated with our program. We have over 90 faculty that range from really amazing but down to earth Nobel laureates to really incredible junior faculty. And regardless of what people's background is, we're all highly committed to training scientists and take graduate student education and undergraduate education very seriously. Our program is very large. We take on average 38 students a year. And in the fall, we have our largest class ever of 50 students. So we're really excited about that. Last year's stipend was 37, and this has increased by several $1,000s. So your tuition and benefits and a stipend is provided throughout the entire time you're at UC Berkeley. And on average, that's about 5.5 years. And we train you for a wide range of careers. We give you writing and presentation skill set building, career development. We have an industry liaison program and official networking system, as well as for computational biology and genomics. And I also want to say that we have a very diverse group of students who are very interested in creating an inclusive environment. And IMCB is a group that I'm really proud to be affiliated with, as it's a graduate student-led initiative to ensure that the faculty and staff are doing everything they can to have an equal playing field, to give equal opportunities, and so that everyone has the potential in our program to achieve all of their academic, scientific, and career goals. And generally you get to hear about what the faculty do. You do rotations to find that perfect fit of scientific and mentoring. You learn how to teach, you do presentations, you do your qualifying exam, and the bulk of the PhD is spent doing your research and preparing your dissertation. And again, research is divided across all of these different disciplines, and that all of the graduate students, and this is the picture from my lab right before the pandemic, the last time we had a group meal. I just want to stress that all of the graduate students in our program pursue unique research projects in the lab that they're in. And so you learn how to take ownership of that and become a very rigorous scientist while pursuing unique, important questions in your field. So if you have questions, this is the link to our grad program. Oops. This is my email. Hannah Bloom is the head graduate student advisor, who's a great resource. And Karina Galicia is the director of student services. They're both really wonderful. And if you're interested, we hold a variety of workshops throughout the year about how to apply to Berkeley. And also, once you're invited for an interview, what to expect in the interview, and where you get to interface with students and faculty who are in charge of these, and can give you a lot of great information so that there's no surprises. With that, I'd like to just tell you a little bit about what my lab does, and later I'm happy to take any questions about the program. As Carmen mentioned, my lab studies the molecular and cellular mechanisms underlying our sensations of itch, touch, and pain. And we use a variety of different model systems and approaches to understand these basic senses. Now, Aristotle was the first to define the 5 senses that allow us to interact with the world. But unlike hearing or vision or taste, we know surprisingly little about our sense of touch. And to me, this is so surprising because touch is important for everything that we do. So touch sensitive neurons, we know, are important for emotional well-being. I think during this pandemic, that loss of physical contact with our friends and family has been really emotionally debilitating for all of us. The bond between a parent and child is first forged through touch. We know that touch is important for normal brain development. So premature babies in incubators have a regular schedule of tactile interaction with family and loved ones to ensure that proper development. Touch sensitive neurons are also important for everyday tasks. They mediate our ability to walk in a straight line without staring at our feet, our ability to learn to read Braille or play the piano, texting, all of these things that we take for granted. And altered tactile sensitivity is the #1 complaint in the clinic. And there's a range of disorders, including repetitive use injuries that cause numbing and tingling, tissue injury, cancer. Diabetic neuropathy can lead to repetitive injuries due to a loss of sensation and is the actual reason for amputation. So it's a consequence of this loss of tactile sensitivity, not diabetes itself that causes amputation. And also chronic itch, and maybe as we've been wearing masks all the time, maybe we've adapted a bit, but I know a lot of people have been complaining about itchiness from that as well. We know that pain can be protective or destructive. So acute pain is an important warning system that helps us avoid stimuli that in our environment that could cause tissue damage. Inflammatory pain is a protective system. So if you get a burn, that hypersensitivity allows you to take some time and protect that area to promote healing. But chronic pain serves no purpose and doesn't go away when that injury or disease is gone. And chronic pain and loss of pain are equally debilitating. And if you're interested in this topic, there's some really great articles in the popular press. On the one hand, loss of pain leads to congenital inability to detect pain. And pictured there is Ashlyn Blocker, who had broken bones and didn't even notice when she was playing basketball, or when she was cooking, she was stirring pasta, and when the spoon fell into the boiling water, she would stick her hand in the boiling water and not even feel a thing, and so was constantly injuring herself and causing tissue damage without feeling any pain sensations. And at the other end of the spectrum, here's a picture of Cameron Bosdog, who suffered from a burning pain syndrome, where in response to even small movements or slight sweating during activities, her skin would feel like it's on fire. And so we're interested in conditions like these, what triggers them, what's gone wrong in the system. And chronic pain affects more than 10 million people just in the United States alone. There are no cures, and the treatments that are available only partially alleviates the suffering in most of the patients. And of course, there are many side effects of the current treatments out there, including addiction and tolerance. So the challenge is to discover genes that underlie chronic pain so that we could develop new therapies that specifically target pain at its source that might be without these terrible side effects. So to do this, my lab studies the somatosensory system, which are clusters of neurons shown here in green in the human on the right in that cartoon, and also in this mouse embryo where you can see that all of the somatosensory neurons have been labeled in green. And these neurons, these millions of neurons that are innervating throughout your body, mediate environmental stimuli, including pressure, both noxious and innocuous, temperature, as well as chemicals. So if you squirt lemon in your eye. That pain you feel is through activating the somatosensory system, but these neurons are also sensitive to internal chemicals that are produced under tissue injury or disease conditions. There are different subsets of somatosensory neurons that mediate different sensations, and they are roughly grouped into two different categories, including nociceptors that transmit pain signals from the periphery to the spinal cord and eventually to the brain, and touch receptors that mediate gentle touch, and proprioception or awareness of limb position that also then transmit signals from the periphery to the central nervous system. And generally there's two clusters, the trigeminal ganglia cells that innervate the head and face and mediate things like eye pain, headache, texture, preferences among food, and the dorsal ganglia that are the neurons that do the same function but for the rest of the body. So the questions that we're trying to understand include addressing these questions: how does force excite sensory neurons? How are mechanical pain thresholds set so that we could distinguish between the brush of a feather or the prick of a pin? And how does injury or disease lead to altered tactile sensitivity, such that someone who suffers from chronic pain feels the feather as incredibly painful? Somebody with diabetic neuropathy might step on a tack and not feel it at all. And somebody who suffers from chronic itch might find the pain of painful stimuli, like scratching your skin really hard, as pleasurable. What happens in our nervous system that allows these switches in how we perceive the same stimulus change under different conditions? And so I'll just tell you about some of our work on itch, which is described as an irritating sensation that causes a desire to scratch. I think you've all experienced this from a mosquito bite or poison ivy, or from your mask, and maybe came up with creative ways to scratch that itch as well. Under normal conditions, itch is thought to be a protective function that warns us about those malaria carrying mosquitoes and protects us from burrowing insects, from burrowing deep into our skin. It also teaches us behaviors to cover up or put on DEET when there's a lot of mosquitoes around. And it's also highly conserved. So not only is it in mammals like humans or lions, but fish also have behaviors to remove irritating parasites, like rubbing against coral in this case, or visiting a cleaning station in the case of this eel, where other animals remove the parasite from them. And even Drosophila have an itch-like behavior to remove mites from their skin. Oops. But chronic itch is very different. So if you imagine the very worst itch you ever experienced and what life might be like if that itch were to spread over your entire body and if it were present for every second of every day. And chronic itch is just as debilitating as the very worst pain syndromes. And 1 in 10 people will suffer from chronic itch at some point in their lifetime. It could be from allergies or skin conditions like atopic dermatitis. Some cancers like lymphoma have really horrible chronic itch as a side effect. Infectious diseases can cause chronic itch. There's a neuropathic form of chronic itch that's due to nerve damage. And there's a growing list of drugs that cause chronic itch as a side effect. And it's thought that in all of these cases, there are very different mechanisms that trigger chronic itch syndromes. But the one thing they all have in common is that antihistamines are not effective in treating chronic itch. And actually, in some cases, they make the situation much worse. And there are very few effective treatments. So there's a lot of interest in understanding mechanisms underlying chronic itch. And so my lab has spent the last 8 years or so trying to understand the cells and the signaling molecules that drive chronic itch, as well as different chronic pain and inflammatory disorders. And so I'll tell you just a little bit about what some of the projects are in my lab that graduate students have initiated. When I was a postdoc and was just starting my lab, I never thought that we would study chronic itch, but I had two very talented graduate students join the lab, Sarah Wilson and Takeshi Morita, who decided that that was a question that they wanted to tackle. One of the models that we've been using is to try to understand atopic dermatitis or eczema, which is the most common chronic itch disorder. And they developed different mouse models of atopic dermatitis to try to identify neuronal mechanisms as well as neuroimmune mechanisms that drive this really chronic disease that's characterized by red, itchy, scaly skin. There's an infant form of eczema, but also it's present in adults at certain hotspots where your arm bends and on the knees and also on the neck and face. And the number one complaint from patients is that it's so itchy that it makes it hard to focus on schoolwork or go to your job or to sleep. And in children especially, there's high levels of depression due to the chronic itch, and also from bullying and other kids making fun of them. Even in people who have eczema as a child, if it goes away, eczema is also associated with several other diseases. And so there's something called the atopic march, where 70% of patients with severe eczema go on to develop food allergies, allergic rhinitis, and later in life, a severe form of asthma that's very difficult to treat. And so we thought that this would be a really great model to try to study mechanisms of chronic itch from a neuroimmune perspective. And Rose Hill and Carolyn Walsh, they decided to look at the interactions between the immune system as well as the nervous system to try to understand how atopic dermatitis is initiated and how then the sensory neurons contribute to this atopic march to asthma. And they discovered that that there's this very close relationship between the immune system and the sensory nervous system, in that the neurons, upon activation, release a variety of inflammatory mediators and neuropeptides that drive immune cell function and recruitment to the skin. And then in turn, these immune systems release inflammatory cytokines and other molecules that make the sensory nervous system hyperactive and fire very robust action potentials and trigger plasticity at both the peripheral level and the central nervous system, such that even though there's nothing on the skin, the nervous system is overactive and leads to chronic itch sensations. Another model that we've developed in the lab is to develop mouse models to study acute and chronic pain. And this includes looking at multiple levels, looking at the primary afferent sensory neurons that innervate the skin and the gut and the lung, looking at the level of the cell bodies in the ganglia themselves, and also the interactions of the primary sensory neurons with the secondary sensory neurons in the spinal cord. And what we found is that not only are the sensory neurons hyperactive, but that at multiple levels in the periphery and in the central nervous system, that there's a variety of non-neuronal cells that play a key role in affecting neuronal excitability. And these include macrophages that come in and sensitize the cell bodies, as well as glial cells, satellite glia in the periphery and microglia and astrocytes become hyperactive at the level of the spinal cord. And these interactions we know are very important for chronic pain and the switch from an acute pain system or nociceptive pain to an inflammatory condition, and also under neuropathic pain conditions that are due to nerve damage. And one inflammatory pain model that we've been working on, and that Kim in my lab, who's our latest graduate student to join, has been looking at chemotherapy-induced peripheral neuropathy. And so about 60% of cancer patients who undergo chemotherapy develop this disorder called CIPN. That includes pain, burning, tingling, numbness, pins and needles, and cold hypersensitivity. Of those 60% of patients who develop this, 40% have this syndrome for the rest of their lives, even after the chemotherapy has stopped and after their cancer has been eradicated. And right now, it's not understood why some people develop CIPN and others don't, and while some people recover once the chemotherapy has ceased and others do not. And that's really what Kim's thesis project is right now, is to determine what makes one individual more susceptible than others. And she's using a variety of different genomics approaches to study this. Again, using mouse models, but we're able to look at human disease genes and how they function within the mouse model. And finally, I'll end with our latest work, which has been pioneered by a graduate student, Sonali Mali, that we've started in the last year due to COVID-19, which is try to understand how peripheral epithelial cell, neuron, and immune cell interactions promote lung inflammation in COVID-19. And there's been a lot of talk about how SARS-CoV-2, the virus that causes the COVID-19 disorder, triggers a cytokine storm and immune cells to become hyperactive in the airways, and that leads to lung inflammation. But what many people don't realize is that our nervous system plays very important roles in lung homeostasis under normal conditions, and that the nervous system plays a key role in regulating breathing rates and other functions of the airways, including sneezing and coughing under disease conditions. And so this includes the trigeminal sensory neurons that I mentioned earlier that innervate the nasal cavity, the dorsal ganglia neurons that innervate the skin and mediate cutaneous pain and itch. Also, at the thoracic level, densely innervate the lungs, and again, regulate breathing and regulate smooth muscle and epithelial cell function. And also the vagal afferents that are also sensory neurons innervate the lung as well. And together, these classes of neurons regulate a diverse array of behaviors of the airways. And what we are trying to understand is how the nervous system contributes to airway inflammation following SARS-CoV-2 infection. And really trying to look at some very basic fundamental questions between the airway epithelial cells, these neurons, and the immune cells. Who gets activated first? Who's infected first? And who's contributing in different ways to the cytokine storm? And one way that we've been doing this is by looking at ion channel activity. So we know that ion channels regulates neuronal excitability, but these ion channels are also, different ion channels also regulate the calcium-dependent release of inflammatory mediators by airway epithelial cells, as well as immune cells in the lung. And interestingly, we didn't think about this before, but as we started reading about SARS-CoV-2 during the early pandemic, we found that coronaviruses express viral ion channels in the infected cells to create a favorable replication environment. And so there's at least 2 ion channels in SARS-CoV-2 genome, 3A and E, that get expressed in the infected cells. And we hypothesized that these ion channels in infected neurons and in infected epithelial cells trigger a switch from a state of homeostasis to a disease state. And so we're trying to understand how these viral ion channels alter epithelial cell physiology. And so we could look at this in vitro and look what happens to calcium signals in these airway epithelial cells that have these ion channels in them. Are they releasing more mediators? Oops. We're trying to understand the mechanisms by which sensory neurons are directly infected by SARS-CoV-2, and the latest human studies show that the trigeminal neurons are some of the most robustly infected cells in the brain. And we want to try to understand how infection of these neurons affects airway function and inflammation, and we've especially been looking at inflammatory lipids that we know lead to hyperexcitability. And finally, because we know very little about these ion channels, we've been characterizing their basic function and also screening for drugs that inhibit viral ion channels in human cells, with the idea that we could find some new blockers to use potentially in our research to understand more about the role of these ion channels in the viral life cycle, and also potentially therapeutically. And we've already identified some interesting looking blockers that we hope some collaborators will test in viral infection assays and mouse models. So hopefully our new studies that we've just started in the last year will provide some novel mechanistic insight into the role of the nervous system in SARS-CoV-2 mediated respiratory distress, and also potentially lead to the identification of new drug targets to inhibit airway inflammation due to SARS-CoV-2 infection and other coronaviruses that have these ion channels in their genome that are capable of infecting human cells. Oops. And so with that, I just want to thank the people who have done this work in the lab. An amazing group of graduate students and undergraduates have contributed to all of these research programs and all of the papers that have come out in the lab. I mentioned we're very interdisciplinary and have a wide range of labs that we collaborate with, from experts in lipid inflammatory mediators and mass spec, immunologists, computational biologists, structural biologists, and fellow physiologists, including my long-term collaborator and Professor Ellen Lumpkin, who I have a joint lab, the Sensory and Epithelial Neuroscience Lab that we co-run at UC Berkeley. So with that, I'd be happy to answer any questions that you may have.
Thank you, Diana. What a wonderful talk. I really appreciate this. And obviously, I always enjoy talks when you integrate, the physiology, the molecular, the behavior, the organismal standpoint. And it's such a wonderful, moment to see all that together.
Thank you.
Sure. And I have, I don't know, questions will show up pretty soon in the chat, but I just wanted to ask you if each has a gender bias. Can women tend to don't mind the poll, women tend to have itch more than men, or in female males, whatever? Do you know that?
Yeah, there are definitely it varies quite a bit, and there are different chronic itch conditions that are more associated with women than men. As we age, there is an increase in chronic itch and dry skin itch, and it's not known why this happens, and women more than men, but over the age of 60, it's aside from sort of heart conditions and blood pressure, chronic itch is like the number 3 complaint, and it's really awful. And unfortunately, it's higher in women, and overall, it's about 30% or higher.
Wow.
So
Okay.
there are some conditions, yeah.
I see, because the only time I had this horrible experience with chronic itch was after I gave birth to my children, but it was a dermatitis localized in the joints and it was all the, I'm sorry, I'm bringing this up, but and it wouldn't go away. So my obstetrician said, well, you need to go to a dermatologist. So I went to I could not sleep. I was nursing twin boys with this itch all over your body, and no one could tell me why I had this problem. And so that will be my last, is hormonal changes. Do you think that also triggers? I imagine, I guess that will be the answer.
Yeah. So there is this horrible pregnancy itch that can occur during pregnancy or after birth. And it is very much related to hormones. But sort of why some people get that and others don't isn't known. And I should say that the dogma in the field 10 years ago was that you really need to treat the immune system to treat itch, target the immune system to treat itch. And now there's this movement like that it's a neurological problem. And so it's been interesting to see how the basic research has changed over the last 10 years and the way of thinking that's starting to evolve in the clinic. But I think, targeting the nervous system for pregnancy itch is going to be, I think, something to look at for sure.
Well, definitely will help, because that was crazy days those were.
It's horrible.
Yes, it was. Well, we have a question here. This is my dear colleague and friend of yours as well, Keith Trujillo. He says, excellent presentation. A unique aspect of COVID is lack of distress at low O2 levels that should evoke distress. This may be an example of intersection peripheral central nervous system. Do you have any ideas about this puzzling symptom?
Yeah, it is very puzzling, and it seems like there's sort of a disconnect between what's happening sort of peripherally and centrally. And it's not really known, right? There's very few studies on the nervous system so far because it's just such early days. And even though we knew the nervous system was infected with SARS-CoV-1, there's very few studies about this. But the trigeminal system is sort of interesting that it's so highly infected. The cell bodies are. The central projections go to the brainstem and are in very close proximity of the central respiratory center. So there might be some modulation there, as well as in the peripheral terminals that innervate the airways that regulate breathing rates. So I don't have an answer, but I think it's a really interesting question.
Yeah, it is. And there's another of our undergraduates, Amanda. She says, considering the role of the nervous system in itch, have you or your collaborators looked in the interactions between psychological stress or anxiety on itching or pain?
Yeah, there's been a lot of interesting psychophysical studies on itch. And also, I mean, on pain as well, but definitely, I had eczema as a kid, and being stressed or having anxiety made it much worse. There's also contagious itch in the sense that it's not from physical contact, but seeing somebody itch can trigger you to itch, just like if you see somebody yawn, and there seems to be these mirror neurons in the brain, just like that get activated, just like when you see someone yawn, you have to yawn. There seems to be that for itch as well. And also some of the images that I showed early on with like the bug on the skin have been used in psychophysical studies. And, most people will scratch just by seeing that. So for some reason, itch is a very powerful stimulus, even if it's not you that's experiencing it, that triggers that desire to scratch. And I think it's a really fascinating subject. We're just starting to learn about the cells and the circuits that mediate acute and chronic itch. But I think now that we have some idea from fMRI and human psychophysical studies, that some of the higher levels that process that information, connecting that to the peripheral pathways that we're starting to understand will be really fascinating.
Yeah, it will be. Another question is, can you explain itch related to lupus?
So, yeah, it's interesting. So, lupus, people typically think about pain and achiness, but there are some conditions and reports of patients with other things like itch. And this is true for a wide range of diseases. In some cases, cancer. In most patients, it can cause pain, cancer pain, but in some people, there's itch. And we don't really know why there are these different consequences on the somatosensory system. It could be, we know that there are very specific immune cells and subsets of neurons that interact in different disease conditions. And it could be that depending on, lupus can trigger many different things, and its exact cause isn't known, but perhaps in different people, there are different repertoires of cells that are driving, that are hyperactive, that are driving these different sensory dysfunction. But again, it's a good question that I don't have an answer to. But I think there are tools to start to try to understand this, and people are thinking about this more and more.
Okay. I have another. Oh, there goes all these questions. All of a sudden, good. And there, there's another student says, my comment is about the importance of somatic pain and the relationship with anxiety that causes to a person. That's what she's making that comment.
Yeah, again, I think we talked a little bit about anxiety.
Mm-hmm.
People who tend to be more anxious or neurotic, like me, tend to scratch faster in psychophysical studies. So maybe, I think there's definitely some priming of the system that might make you more susceptible to itch, or more maybe you have more empathy and makes you scratch more when you see somebody else suffering from itch. But definitely, having anxiety already can make these syndromes worse, but they also trigger anxiety. So it's a real vicious loop, right?
Mm-hmm. Mm-hmm. Then are there persons that do feel itch or do have any associated problems?
Yeah, so there are a number of people who don't feel pain, and but they have not been extensively studied with regard to itch. But there are a number of patients who are now currently being studied for itch that have different inabilities to detect different types of pain. So it's a super interesting question. I heard there are some people who don't feel pain, don't feel itch as well, but I've also heard just unpublished results that maybe it's going to be mixed, and it depends on what the genetic cause of their inability to detect pain is caused by. Some people don't have subsets of neurons, some people have defective channels. It just depends.
Okay. And are the viral ion channels in COVID associated to the loss of taste or smell?
So super interesting. Yeah. So in the olfactory epithelium, there it's thought that the neurons that mediate our sense of smell are not directly infected, but their support cells are, and that can contribute to the lack of the loss of smell.
Right.
And in the taste system, I don't know. I don't remember. But in the trigeminal system there, we think that that's maybe affecting other components. So I've heard anecdotally that people who are infected don't experience the burning heat from chili peppers, and that's a trigeminal, not a taste pathway system. So,
Huh.
but it would be, yeah, interesting to see.
Very, very interesting. And there's another, is there a correlation between restless leg syndrome and itch?
Oh, I don't know. I don't know the answer to that. That's interesting. Yeah, it's an interesting question.
Restless, oh yeah, well, those are the ones where we cannot control our legs and when we sleep, I guess.
Yeah, yeah. Also, yeah, and whether, and there's some data that, loss of sensation, tingling and numbing can contribute to these types of syndromes. That it's not just like, oh, I have pain and now a loss of different aspects of our, peripheral nervous system can lead to different syndromes as well.
Wonderful. Well, I think we answered all the questions and we are already 6 o'clock. And I really want to thank Diana for doing this wonderful presentation and answering all our questions, and also being our last very special seminar of our series. And I really look forward to seeing you again, Diana, somewhere, not if it's not in Woods Hole, so maybe in Puerto Rico when COVID ends, you can come and visit, and we'll take you out to the beach and have a nice dinner on your honor. We'll hope to see you again. Yes, of course. And thank you so much. And thanks everyone for being with us and sticking with us all this semester. I appreciate everyone to being here. And have a great end of the semester and wonderful summer, Diana. You take care of yourself.
All right. Thank you. Thanks everyone for the great questions, and have a wonderful summer.
You too. Take care.
Bye.
Bye. Bye.